Hello everyone, welcome to another video. This is my first teaching video. I'm starting 1 mark series. In this series, basically I cover all the 19 subject, and each subject I guarantee you 1 mark. And when I was studying, most of the videos were like 2 hours, 3 hours. I thought there was a need of shorter videos as well. So yeah, this is I chose biochemistry to be the first video. There's a learning curve for me as well. I really wanted to get into teaching, so let's see if I can teach. This is the first time I'm doing it. So yeah, please do comment, please give me any advice as well. I'll take that. And even if this thing helps out 10 people, I'll continue this for all the 19 subject. If it doesn't, maybe I'll just stop. I'll stick with the advice videos and whatnot. So anyways, let's get started. I've compiled this PDF, which you can get it in the Telegram group, which I haven't created. I'll create it by the end of this video when I upload. So yeah, I'll send it out to you. So let's get started. This is for my sake. 1 2 3. Okay. So yeah, first and foremost, I'll start with glycogen metabolism. A lot of questions can be asked from here as well. In fact, this PDF might get you more than 5 6 marks if possible, but my job is to get you that 1 mark. So if I can live up to that expectation, that's well and good. So okay, what is glycogenesis? Glycogenesis is basically formation of glycogen from glucose. So we'll start the reaction from glucose, and it will get converted to linear chain and eventually into glycogen. Now glucose to linear chain, it happens by an enzyme called glycogen synthase, which is the rate limiting enzyme as well. And for you should know, for any cycle, for any of the reactions, for any of the production of anything, rate limiting enzyme are very important. Most of the time questions are being asked over there, so just by heart those. So in this one, the rate limiting enzyme of glycogenesis is glycogen synthase. Potential question which can be asked in the exam. After the linear change, get converted into the glycogen. This happens by the enzyme called branching enzyme. I'll tell you about branching. Basically what it will take the linear change, it will make it into branches, and it will eventually form glycogen. Now the next process being glycogenolysis, which is basically taking this glycogen, conversion of glycogen into glucose, so it can be used into our bodies. So that is called glycogenolysis. So I don't get the logic, like first you have glucose, you made glycogen, now basically you're taking glycogen and going back to glucose. Just remember like this, it will help you out to remember things. I'll say random things here and there. If it helps you out, it helps you remember during when you're doing question, it's nice. Yeah, so now we have glycogen. Now we need to go into glycogenolysis. But prior to that, about some of the glycogen, it enters into the lysosome. It gets converted into lysosomal glycosidase. Why and how it happens? It happens by an enzyme called acid maltase. Question can be asked on this. Whatever I have highlighted in neon, there will be a potential question asked, was asked in the exam or can be asked in the future. And it's an important enzyme as well. I'll tell you in the future as well when you go into the glycogen storage diseases. Now, where all this glycogenesis is happening, it's happening in the liver and the skeletal muscles, where basically in the cytosol of the liver and skeletal muscles. Same thing with glycogenolysis as well. It's happening in the cytosol of the liver and the skeletal muscles. Now let's get started. Basically, we took this glycogen, we're starting the reactions from here. The first enzyme which does this will be glycogen phosphorylase, which will convert it into limit dextrin. Now this is a very unique enzyme. What it does is basically, if you remember here, we use branching enzyme and it became glycogen. Now this is glycogen, it is a branched molecule, right? Now we're using glycogen phosphorylase. What it does, it kind of breaks down, it kind of breaks down those branches. This is the only enzyme which you can do that, but at the same time it doesn't do it very efficiently. What it does, there are like 1 2 3 4 let's say here, yeah, 1 2 3 4. It will break down only till the fourth bond, so first, second, third and fourth, but eventually the fifth one will have the branch. So that's how it works. It is not able to fully just convert it, the whole branch thing, into a linear chain. It doesn't do that way. So this one basically will play the role like that, and the molecule which will be formed, it will be called limit dextrin. If you see 1 2 3 4, and there is a branch over here, so it basically gives a 4 into 4 molecule, 4,4 molecule called limit dextrin. Now to break down further and make it into linear chain, you need another enzyme called debranching enzyme, which is basically same as the opposite of branching enzyme, and you will end up with something called G1P, or glucose 1-phosphate. And again, this enzyme can also perform the same thing, where it turns linear chain into G1P. What happens, there is a transfer of glycan happens over here in this particular reaction which I mentioned. This is a potential question which can be asked in the exam. Now G1P gets converted into G6P by exam called mutase. It moves the phosphatase from the first position of the glucose to the sixth position of the G1P. So it will become G6P, glucose 6, and now it uses an enzyme called G6 phosphatase and remove the phosphate, and eventually you will be left with glucose. So that was glycogenesis, and this was glycogenolysis. And why do I teach this? These are wherever I put this is potential question, can be asked by heart those very well. Learn the spelling as well if you need it. Why I taught that was because of this particular glycogen storage diseases. They are very commonly asked, and they'll be most likely asked in the exam. It was asked for us as well. So let's get started. The mnemonic to remember this was so my teacher was Dr. Azam, right? So it was one pomp, Corey, and make her. So I remember it like that. So we have 6 diseases, 6 glycogen storage diseases like these. So the first one, it is the deficiency of an enzyme called glucose 6 phosphatase, which is basically here. And this particular enzyme deficiency can cause the disease called von Georg disease. One of the most reported most repeated glycogen storage diseases. It has been asked on multiple occasions, and most probably will be asked again. Now the organ involved with this storage disease is liver, and I have also put down the clinical features which are mentioned over here. You will see in the question stem severe hypoglycemia. You'll be see the mention of lactic acidosis. You'll see another thing will be spleen will not be enlarged compared to the remaining these three glycogen storage diseases where the spleen is enlarged. So this can pretty much help you out to distinguish between the different diseases. So yeah, why there is lactic acidosis? If you will know gluconeogenesis, I hope you have studied this before. What happens is glucose 6 phosphate, you will remember it is there over there, which eventually gets converted into lactate. So since this enzyme is not there in this glycogen storage disease, there will be deposition of this, which will enter into gluconeogenesis, and it will lead to formation of lactate. So therefore in von Georg you will have lactic acidosis as well. Then pyruvate can also get converted into acetyl-CoA and eventually fatty acid, which can lead to hyperlipidemia. There will be also ketosis. Learn this by heart. This how you should know is you should know the name, you should know the enzyme deficient, and you should also try to remember the clinical features first. These three things you should try to remember first. So most of the questions are based around it, and if you can remember extra like organ involved and 1, 2, 3, 4, that'll be like more than enough. Then the next disease which I'm going to talk about is Pompey's disease. This is in which, if you remember in lysosome, I taught you this enzyme called acid maltase. If this is deficient, this causes disease called Pompey's disease or Pom's disease. And then we have the third one. It is Cory's and Anderson. There's a mnemonic for it. It's called ABCD. ABCD. So A for Anderson, B for branching, C for Cory's, and D for debranching. So what you can do from here is basically, if it is Anderson's disease, the enzyme deficient is branching. And if it's Cory's disease, the enzyme which is deficient, it is debranching enzyme. So yeah, first learn the name, then learn the enzyme which is deficient, and then over these three diseases, they have similar clinical features which can be used to distinguish between von Georg in a way that spleen is enlarged here. There won't be any lactic acidosis you will see over here. So yeah, then the mnemonic one pump Cory and make her tired. So let's do with make, which is McArdle's disease. In this one, most probably in the stem, the thing which will be mentioned will be about a patient or anyone who's doing exercise, is not having tolerance to an exercise, is having muscle cramp. It's mainly happening because of deposition of excess lactic acid during the exercise. So that will be most likely the answer will be McArdle, or you'll have to choose the clinical feature, or you'll have to choose the enzyme deficient, which is glycogen phosphorylase. Also glycogen phosphorylase skeletal. Okay, it can also be in hepatic. If that is the case, then it will be Hirsch's disease. And this one, the organ involved will be liver, and it will have hypoglycemia and hepatomegaly. Most likely, I'm hoping von Georg is asked again, and or it could be one of these. And I think these most likely among these four, the question will be asked. So that's get done. Now we're moving into the glucose transporters. Very important topic. It integrates with physiology as well. So I'm gonna move forward with that. So sorry. So yeah, GLUT1 and 3, they are present in most of the tissues on the body. By the way, this is my handwriting. This is the note I was using during my preparation. So I just took the screenshot and I put it there. So this is your handwriting. Thank you so much. Please don't get mad. Don't copyright me. I don't know if you can copyright someone in notes. Can you? Also, yeah, my teacher was Dr. Azam. So in a way you can say it's his notes as well. And his way of teaching as well. But I don't think they can call it's a good thing, right? I'm teaching you guys forward. Anyways, I'm wasting time now. Let's get started. I want to end it in 30 minutes. Okay, now we're into glucose transporters. You need to know the name, numbers first and oh shit, what am I doing? Yeah, first and foremost, what you need to know is you need to know the name 1, 2, 3, 4, 5, and then where they are present. Very important. And features question can be asked, but first and foremost, I think the numbers and the location which are present over there. So one is there in brain, RBCs, placenta, retina. GLUT3 is present in neuron, placenta, kidney. And the feature of 1 and 3, you can kind of say they're involved with basal uptake of the glucose. If you remember, if you see, like, these are very important things, RBC, placenta, like if the glucose goes, like you're pretty much done. Like neurons are very important part of our body, so they are trained in such a way that even they can take or uptake glucose when there is the body is in the state of hypoglycemia. Now, GLUT2 are there in liver, pancreas, beta cells, kidney, and PCT. Their features is entry and exit of glucose. GLUT4 are present in muscles, adipocytes, cardiac muscles. And I think this question has been asked. In the feature of these four, GLUT4 is insulin-dependent transporter. GLUT5, sperm cells, enterocyte, and testes. I think this question was asked for me. I don't remember exactly, but the feature is it transports fructose. I always remember that sperm, they like something sweet, fructose is sweet, so try to remember like that. Another question which can be asked is that where is the decrease in the glucose transporters? If you're fasting, after overnight fasting, the glucose transporters, there will be decrease in adipocyte. This will be the most likely answer over there. Now there is SGLT1 and 2. One is present in intestine, 2 is present in the lumen of the kidney. It absorbs glucose. There's the other one, or number 2, is involved with reabsorption glucose. I'm sorry, the thing is that I'm trying to teach fast because my first idea was to finish this video in 15 minutes, but when I compiled the document I realized it's not possible. So yeah, so I also added something from LMR of Dr. Azam which can be helpful for you. You can find this PDF most likely in my Telegram group, so don't worry about this. Okay, now next, B complexes questions are being asked in vitamin. Learn the vitamin bit, either you find in vitamin C, I'm saying PSM, or anywhere, wherever you can, go for it. So how to learn this and what's the most important thing to learn in these initially? First, you know the B1 to B12, whatever. Then make sure you know the name. B1 is thymine, B2 is riboflavin, B3 is niacin, B5 is pentathionic, B6 blah blah blah. Learn the name off with the numbering B1, B2, B3. Okay, that's going to be very important for you. Then coenzyme enzyme also you can, but check that for now. Then you know what disease it is associated with. If you have done these 1, 2, 3 things, most likely you will be able to solve most of the questions which come from that. If you're unlucky, they can ask something about coenzyme and enzyme, but I think eventually you should start with that. So thymine, name B1, thymine associated with beriberi, also associated with Wernicke-Korsakoff syndrome. This is associated with people who drink. The mnemonic was Goa, G for global confusion, OA, I forgot. So look into that, you will find it in your psychiatry notes, I believe. Then yeah, so basically with beriberi and Wernicke-Korsakoff. Second one, riboflavin associated with chylosis, stomatitis, and glossitis. The question stem will most likely mention something like tingling of the mouth, tingling sensation, mouth fissure, decrease in glutathione reductase. Most likely, if that is mentioned, the thing answer will be one of these, or it will be this, or it will be this. Then we have B3 niacin. It's a very important thing because almost every time question is asked about about pellagra, and learn the picture of pellagra. It has that Kiesel's necklace as well, and also something about RBC ketolase, which is used to determine, or it's a test, or like it decreases. I'm not very sure about it, but if you know this, you're pretty much sorted. B5, it is pentathionic acid. Penta means 5, B5 means 5. We have 5 fingers, we have 5 toes, so it is associated with foot, burning foot syndrome. B6, pyridoxine, sideroblastic anemia. B7, biotin, associated with alopecia. So there's also another way where biotin can get deficient. Question can be asked that someone is consuming raw eggs. So raw eggs have this component called avidin. It is present in the egg white, not the yolk part, which can eventually deplete biotin in your body, which can eventually lead to alopecia associated with biotin B7. Cobalamin, megaloblastic anemia and peripheral neuropathy. Folic acid associated with megaloblastic anemia and neural tube defects. Ascorbic acid, vitamin C associated with scurvy. Most likely this will be the answer. Also there are X-ray involved with this. Open your pediatrics and learn scurvy properly. Another question which can be asked with vitamin C or ascorbic acid is that they might ask which one is associated with or related with post-translational modification of collagen. It will be vitamin C. It is a good antioxidant and also helps in absorption of Fe. All right, now we are towards I'm gonna get into protein metabolism. A lot of questions get asked over here, so let's get started. I already see. I'm not gonna go in depth, and this is the first video as well, and I do not think I should, because I'm not teaching the whole subject. I can do that. It's not a problem. But this is a short one. I want to make sure that if you watch this in 2 minutes, it's about 10 to 15 minutes video, and you can pretty much go on with your day. Watch this while you're eating or something. So I already assume that you already know the classification of protein metabolism. Learn it because questions do come in it. But I'll start with just getting into the question, the things which can be asked or were asked previously. So first and foremost, the questions which can be asked about glycine is about facts of glycine, which is it is the simplest and smallest. It is the most abundant in collagen protein. It is very flexible. That's why it binds with protein chain because of its flexibility. Okay, this can be asked potentially, or it will ask what is the function or what does it make. So first, then you learn it is involved production with heme synthesis, purine formation, glutathione formation, creatine formation, acts as a neurotransmitter as well, and also associated with conjugation of bile acid. These 6-7 things you learn that. After that, moving into glutathione and creatine formation, what you can do is, since you know that glycine is used to form glutathione or creatine, you should learn the component which is used. For glutathione, it's a tripeptide. Similarly for creatine as well, I remember creatine formation as GAM, like game, gaming, GAMnine. That's what the mnemonic I use. So GME, if you tell me GME, I would remember glycine, arginine, and methionine. It just comes to me. Creatine like that. And glutathione, I remember it as GCG. So I will remember that one of the G is glycine, then this one is glutamate or glutamic acid. The middle one will be cysteine. Here, the neon bit, most likely the question can be asked from here. Most likely I'm expecting. It can asked even from purine, of course, but I'm just telling this is the most likely bit. Now moving into alanine, just learn it in Cahill cycle. Now branch chain, we have valine, leucine and isoleucine. Valine, leucine, isoleucine, it gets converted into a certain product. You don't have to go into depth, but the enzyme which is used to convert this into a product is called BCKD. Or Buckard. I remember it as Buckard. So learn the full form, because in the exam you won't be given short forms for almost anything. So it will be a branch chain keto acid dehydrogenase. So what happens if this particular enzyme is deficient? It result into a disease, or these components will be deposited in the urine, and the urine will have a very specific smell called maple syrup urine. Smell. The urine will smell like maple syrup. What is maple syrup? Like the tree is very popular in Canada, and also branch helps me out. So when I think about branch, I think about these things. I think about tree, and I think about maple tree. Think about Canada. So yeah, the clinical feature is in blue here. I have highlighted it. The urine smells like maple syrup or burnt sugar. Basically that. Okay, then. Another note which I have made down: if they ask what's the enzyme deficient, the answer will be Buckard. If they ask what is the defect in the mechanism, it will be decarboxylase. Okay, moving forward, now we finished glycine and branch chain. Now we're moving into aromatic, because these were aliphatic above. These are aromatic now. The one which have smell aromatic will be tyrosine, tryptophan, and phenylalanine. Tryptophan in turn gives into another melatonin, serotonin, which I'll give into depth now. So tryptophan gives rise to, or it can be used in production of 3 things: melatonin, serotonin, and vitamin B3. Niacin, if you remember, I just taught you before, which is associated with pellagra, also Yeah, pellagra mainly. So yeah, let's get started. And when I say pellagra, vitamin B3, this is one of the only things which can be converted into a vitamin, which can be a potential question which can be asked during the exam, which among this can be converted into a vitamin. So since I mentioned it can be converted into melatonin, serotonin, and vitamin B3. And melatonin is associated with the sleep cycle. Serotonin, it has a lot of other things, but one question which can be asked is the clinical correlation is carcinoid syndrome, in which 5-HT, which is 5-hydroxytryptamine, also called serotonin, is high. The clinical features will be flushing, endocardial fibrosis, diarrhea, tricuspid insufficiency, which will basically give you a hint about carcinoid syndrome. Either answer will be that or answer will be this. And then, as I said, this can be converted to niacin, right? So if there is a deficiency of tryptophan, the disease which can be caused is called Hartnupp's disease. And it makes sense, since it makes niacin or vitamin B3, what you realize is that symptom of this particular disease, Hartnupp's disease, will be something similar like pellagra, because it makes niacin, and niacin is, you know, it causes pellagra. And if this particular thing is deficient, it causing a disease called Hartnupp's disease, the symptoms will be something similar to pellagra. Okay, moving forward, we move into phenylalanine and tyrosine. So let's look into this particular reaction where phenylalanine gets converted into tyrosine by an enzyme called phenylalanine hydroxylase. Meanwhile, on the side, there's something called BH4. 4 means tetra, 4 means char. Tetrahydrobiopterin gets converted into BH2, which is dihydrobiopterin. So in this particular thing, the question which can be asked is that if this particular enzyme tetrahydrobiopterin is deficient, the deficiency of this particular thing can lead to enzyme a disorder called Sagawa syndrome. Most likely answer will be this, or the option will be this. Okay, moving forward, as I said, it gets converted into phenylalanine. Now let's say if this enzyme is deficient. If this enzyme is deficient, matlab, it will not be formed. If this is not formed, what will be accumulated or increase in value? It will be PA, or phenylalanine will be increased. And now if that gets deposited into the body, into the urine, it causes a disease called phenylketonuria. Not urine, but generally I'm telling in the body. This then I have put down the blue color. I put down highlighted the clinical features. The person will have pale skin or fair skin. I'll tell you this moving forward why they will have pale skin. They'll have blonde hair. They'll same thing due to pigmentation issue. It's not melatonin, it's melanin. Then they'll have mental retardation. They'll have musty odor or the smell because of phenylalanine. As I said, that these are aromatic compounds. That means they have their own particular smell. Another question which can be asked, it can be why and I mean how it is tested. The test of this is called FeCl3. I'm not sure, but I think the other name of this FeCl3 test is called Guthrie test. So have a look into that. Now tyrosine, the same thing. Tyrosine can also get converted into melanin by an enzyme called tyrosinase. So yeah, so basically if tyrosine is not formed here because of this enzyme in phenylketonuria, melanin will also not be formed. That's why these people have fair skin and they have blonde hair. I hope that makes sense to you. I gave the logic of why they have fair and pale skin. Now we're getting to this reaction. Melanin is being formed by the enzyme called tyrosinase. Now if tyrosinase is deficient, matlab melanin won't be formed. If melanin is not formed, it is associated with pigmentation of our body, so it can lead to potentially albinism and vitiligo. Okay, now into T3, T4. If you have done medicine, this will be taught there, so I don't see the need of teaching this, and it will be integrated, so I don't think I want to go into much more depth into formation of T3 and T4, but yeah, tyrosine is involved over there. Now get into phenylalanine and tyrosine can also be used to make catecholamines. Catecholamines would be dopamine, epinephrine, and norepinephrine. So we already know this particular reaction, phenylalanine to tyrosine. It happens how? It happens by PA hydroxylase. Now tyrosine can go into melanin route and whatever route, but in this one time, this will go into the next route called dopa, in which the enzyme will be used is hydroxylase, same as here where the water is getting added. Water OH is getting added, and it will form dopa. This particular thing is a rate limiting enzyme. Potentially question can be asked, what is the rate limiting enzyme over here? Then it uses decarboxylase. Decarboxylase, D means remove carbon from there, so carbon will go out and it will form dopamine. Now we just have to make norepinephrine and epinephrine. So remember like that. We use hydroxylase, we add water to dopamine, it will become norepinephrine. And then norepinephrine is called norepinephrine because the methyl group is in a different location. We just have to move the methyl group from certain position to another position, and that will happen by an enzyme called methyl transferase, and it will eventually, the end result will be epinephrine. Meanwhile, what happened? SAM, the full form of SAM is S-adenosine methionine, gets converted into S-adenosyl homocysteine, in which this is the donor, and methyl group gets transferred, and eventually norepinephrine gets converted into epinephrine. Alright, now we are towards the end of this video. It's turning 25 minutes. I think in 5 minutes I should be wrap this up. So yeah, now again tyrosine degradation, in which there's a certain reaction. You don't need to learn it. What you need to know is homogentisate gets converted into blah 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 by an enzyme called homogentisate oxidase. Now if this particular thing is deficient, what will happen is that this will accumulate. Homogentisate will accumulate in the body. It can accumulate in the connective tissue, it can accumulate in the urine, and the most likely the question which gets asked over there is, over in this particular thing, is that when the urine gets turned black when it was left standing. And the disease I'm talking about is called alkaptonuria. These are the clinical features which I have mentioned in blue color. What is ochronosis? Ochronosis is basically the same thing, deposition of the same thing into the connective tissue or the tissues. It gives a black discoloration. I mean, if it can give black discoloration to the urine, it can give the same thing to the tissues as well. Also, it can cause calcification of vertebrae in the x-ray as well. So there is that. I have added this table, whatever I was taught before, because this will be quick for you to remember. This question is very commonly asked: what makes nitrogen oxide? Answer is arginine. And also arginine also makes creatine. If you remember, GAM, this was glycine and this was arginine, this was methionine. So yeah, tryptophan, I told what all it does, melatonin, serotonin. So this table can potentially help you out. Now another important topic, Leach-Nyhan topic, Leach-Nyhan syndrome topic. This came in the exam. Learn the full form of HGPRT. I forgot because every time I see it, I remember they're talking about this enzyme. So fine, Google it, type HGPRT, you'll pretty much know. I think most likely they will mention the short form, hopefully. But the very specific, I mean, there is these three clinical features which are commonly asked and can be asked, but the most important one is this particular one, which is self-mutilation. So this enzyme is deficient. It will lead to a syndrome called Leach-Nyhan syndrome. These will be the clinical features, and they can potentially even put a picture of a child or anyone who has like bitten off his lips, or has self-mutilate themselves. So yep, there is Leach-Nyhan syndrome. Then talking about gout, it's in Purines. So not to go into much deep, but maybe just remember this much. Even this is not very important, but if you know gout, why it happens? It happens due to deposition of uric acid or urate crystals in your joints. All right. So what potential? So this particular gout thing, it integrates with pharma as well. So I'm telling the pharma integration over here. So if you know the drug of choice, if you've done medicine, you would know the drug of choice for, not drug of choice, or drug, one of the drugs which is used is xanthine oxidase inhibitors, and the name is allopurinol, right? So this is basically the reaction, hypoxanthine into xanthine and eventually the uric acid. So this happens from here to here. Eventually this, which gets accumulated in the joint and causes gout, is done by these two enzymes. Now if you get something like an inhibitor, something called allopurinol, which is an xanthine oxidase inhibitor, if this gets inhibited, this will not work, and eventually there won't be any uric acid, and eventually there won't be any gout. So that's the logic behind it. So that's how it can get integrated. I have put down Leach-Nyhan syndrome, HGPRT, what all it happens. Then we have gout, it causes hyperuricemia. These are the crystals which are present in the thing. So yeah, learn the pictures of these needle shaped structures. There are of two types and all that. So you'll have to open nephrology for it in your medicine. So yeah, that was a video. I thought I'll finish in 15-20 minutes, but damn, it became 30 minutes, and I don't know how these teachers teach for 7-8 hours on a day. It's crazy to me. But I hope this was helpful. The whole point is that I'm able to get you that 1 mark, but whatever I taught you, definitely it covers like nice 5 6 marks, maybe most of the things. Also another thing, you can do ETEC, electron transport chain, which definitely guarantees you 1 mark as well. So if you comment down below, maybe I can teach that just that one topic in depth properly so you can remember that. Hi, so I was saying that, yeah, that was the end of the video, and please do comment because I really want to know how I did. Well, I did not go into that much depth, and also the subject you cannot go into the conceptual knowledge because it's biochemistry. It's pretty much very factual, it's very straightforward. So let me know how I did and if I should continue doing this, because even if it helps out 10 people, I'll be happy to make it for the remaining 18 subject as well, because uh, I wanna I wanna get you guys at 1 mark in each subject, and you'll also get an idea about what are the important topics. But whatever I've compiled, most likely it gets you about 6 7 marks. So there is that, and I can teach in more depth, but I don't want to make that long videos, because when I was studying, I wanted to see some short 20 minutes, 30 minutes video where I can just watch into 2 into speed, and I get like the maximum value out of my time. So yeah, do let me know, and you'll find this PDF. And yeah, so this is the first, like these video take time because I have to first figure out what I need to put into it, and also these are not everything is my notes, so I'm a bit scared that some people can complain about it. They shouldn't. Hopefully they shouldn't. And if I sit down and write, that's gonna take me a lot of time. So yeah, let's see. Please do comment if I should continue doing more of these videos, and or if I should just teach the whole subject, if I can potentially can. And yeah, until next time, I will definitely make more advice videos, but I really want to get into the the teaching bit, so I really hope it will, like, it helped you out. Thank you so much. Thank you so much for watching. Until next time, bye bye.